translation, specifically the three phases: initiation, elongation, and termination. I also have a video showing some of the process of elongation without all the details that you can take a look at on PolyLearn. So in this particular slide, you're looking at the formation of the 30S initiation complex, which begins with IF3 binding to the 30S ribosomal subunit. And that step is essential to keep the 50S subunit from prematurely binding and making a little ribosome sandwich before the messenger RNA can bind. Off to the left here, you see initiation factor 2 binding GTP and then facilitating the insertion of the FMET tRNA to the initiation complex. And so that's all here. So you've got initiation factor 2, GTP, and then the FMET tRNA forming a complex with the 30S ribosomal subunit. As the 50S subunit binds to the 30S subunit, GTP is hydrolyzed, IF1 and IF2 are released, and the 70S initiation complex is formed. And there's our entire 70S complex. The 50 plus the 30 non-intuitively gives the 70S with the messenger RNA and the tRNA FMET in the P site. Now we have an empty A site ready to receive an incoming tRNA. So again, the elongation cycle consists of three specific phases: the aminoacyl tRNA binding, the transpeptidation reaction, and translocation. And that's all highlighted here. So here's our complex. The empty A site is illustrated, the P site is illustrated, and the E site is illustrated. Over here, you've got a cycling reaction with elongation factor Tu and elongation factor Ts that facilitate addition of amino acids to the growing chain and work towards facilitating the transpeptidation reaction with elongation factor G and GTP. So as we see here, the aminoacyl tRNA GTP EFTU complex, which is pictured here, brings the aminoacyl tRNA into the A site right here. So again, follow the little yellow amino acid that enters the A site. The process then, you see a dephosphorylation of GTP resulting in leaving behind GDP on the EFTU. EFTS comes along to kick off the GDP from EFTU, and now GTP can bind to EFTS and renew this energy generating process that allows this complex to grab the next aminoacyl tRNA and throw that into the empty A site as this complex moves. The transpeptidase reaction is pictured here, where you're seeing elongation factor G form a GTP complex and facilitate translocation of this chain here. These two amino acids get added to this amino acid, and that's what's pictured here. And of course, the ribosome slides over. And that process, as I said, is facilitated by elongation factor G and the hydrolysis of GDP. And of course, at the end is termination, where you have various release factors that work together. And so in this particular case, we see termination. We reach a stop codon in the messenger RNA, and release factors 1, 2, and 3 are added sequentially. And so again, these release factors bind and hydrolyze GTP and facilitate the release of the ribosomal subunits. Initiation factor 3 can then bind back onto the 30S to prevent 50S ribosomal formation of the 70S complex. And again, going back to the concept of throwing antibiotics in whenever I can and just making this a little bit more practical. This is the general mechanisms of protein synthesis inhibitors, and so just a few different classes of antibiotics that exploit the essential ribosomal function. So macrolides, for example, antibiotics that bind to the 50S ribosomal subunit, and typically they'll prevent transpeptidation and continuation of protein synthesis. There are amino acids that there are aminoglycosides that interfere with the 30S ribosomal subunit and cause misreading, so that in a sense, they distort the A site, for example, and disallow amino acyl tRNAs from inserting their amino acids into the A site. Tetracyclines also block the 30S ribosomal subunit. So again, interfering with tRNAs moving in. Streptogramins are also another class of antibiotics. I put these little notes down here just so you can kind of see. I'm not going to ask you exam questions about that, but streptogramins, for example, interfere with peptidyl transferase, that reaction that I showed you here. This is the peptidyl transferase reaction that's happening. So these two amino acids, when they get moved over onto that to form this, is caused by the 50S ribosomal subunit, specifically the 23S ribosomal RNA in the 50S ribosomal subunit that facilitates that reaction. So obviously antibiotics like macrolides and so forth that interfere with 23S ribosomal RNA activity in the 50S ribosome will interfere with that type of transpeptidation reaction. Anyway, you don't need to know all these. I just wanted to put that in there. Well, actually, I do want you to know the mechanism of action of aminoglycosides just binding to the 30S, and tetracyclines binding to the 30S, and lastly, macrolides binding to the 50S. Another thing that has to happen, though, is once the polypeptide is synthesized, it has to be folded typically, and often it's got to be folded in the right way so that it can be functional or secreted. And so this slide illustrates that point, where you initiate with chaperone proteins like DNA-J that binds to the unfolded or partially folded protein. So here's a dimer of basically, well, it's not a dimer, but two DNA-J proteins interacting with DNA-K, who in turn is bound by ATP. And here's our unfolded protein. So there's obviously a hydrolysis of ATP. You see the two inorganic phosphates being released, and that's DNA-J stimulates that ATP hydrolysis by DNA-K. So DNA-K is the actual ATP hydrolase, but once DNA-J is bound, that facilitates that catalysis. So we end up with ADPs on each of the individual DNA-Ks. We need an additional protein called GRPE, and in this particular case, there's it's basically a nucleotide exchange factor. It stimulates, basically just kicks off the ADP. And so in this phase right here, the protein is essentially being folded. And so as the protein gets folded, it may be partially folded, so it may have to go back through this and get folded again, to be in its final conformation, or it may get fully folded and then results in its native active conformation.